Good evening, students. The other day we were discussing the lichens, the meaning of lichen, that is an association of algal component and fungal component, and the association is generally called the symbiotic association, but later it may be called as this helotism, the master and servant relationship. And we discussed different types and classification based on the thallus organization and also based on the fungal component. The fungal component is an ascomystis fungus. They are called ascolichens, and it is a basidiomystis component. They are called basidiolichens. And also the vegetative propagules associated with the lichens also we discussed. Today we discuss the reproduction in lichens. So regarding the reproduction, it is general in majority of the species, majority of the plants you can find vegetative, asexual and sexual, but sometimes you can find only asexual and sexual only. Here you can find the three types: vegetative reproduction, sexual reproduction and asexual reproduction. Now let us see the vegetative reproduction. This vegetative reproduction by fragmentation, generally the thallus may be cut into pieces. Each piece is developed into a new thallus. So generally the fragmentation occurs in foliage and fruticose lichens. Some other structures like Isidia, it is a small cartilaginous outgrowth found on the upper surface or upper cortex of the thallus, and it is made up of some algal cells surrounded by some fungal filaments. This type you can find. So this Isidia may be discharged. Here you can find this one called Isidia. There you can find upper side, you can find a cartilaginous outside, a cortex like structure is found, and inside you can find the algae. So it may be detached from this and grow into a new thallus, lichen thallus. This type of vegetative reproduction is found in Parmelia and Asnia. Next, another type is called the Soridia. These are small, non-cortical, bud-like, powdery masses or outgrowths made up of few algal cells. A few algal cells surrounded by only few fungal cells. So a little amount of algae surrounded by fungi and formed a small structures, just like powdery structures found on the thallus. They are called Soridia. Example: Parmelia and Bryophyta. See, look here. Here you can find on the upper surface of the thallus, you can find the Soridia are found. A small amount of algae surrounded by fungal hyphae. They just developed, a bud-like structures developed, and they will be released out and they will develop into new lichen forms. So each Soridium consists of an algal body and surrounded by fungal filaments. The next type reproduction, the lichens are reproduced asexually by forming the following fungal spores. The following, here you can find mainly the asexual reproduction by the fungal origin only. This is F, fungal origin. That is, algae, there is no chance of reproduction in lichen forms. That's why the appropriate word is helotism, because the reproduction, whether it is asexual reproduction or sexual reproduction, it is carried out by the fungal component only. Here the spores, here you can find the oidea. The oidea is nothing but small pieces that you can learn in fungi also. The hyphae or the filament may be break into small pieces, and each piece is becomes circular and form into a spore oidea, and it will develop into a new mycelia. The next type, the conidia, this also you know in fungi. The conidia, there you can find these fungal component of the lichen forms conidiophores. Conidiophore means, I told you already, 4 PHO-REO4 means a stalk-like structure. So at the end of the stalk-like structure, you can find chains of cells formed these conidia, and they may be removed and develop into a particular fungus. This type you can find in Roxella and Cladonia. This you can see. So here you see this is a fungal filament. In the fungal filament you can have a uninucleate filament, where you can find at the tip of this conidiophore stalk you can find chains of conidia are developed. When the conidiospores mature, they will be removed and find on a suitable substratum, they will develop into that particular fungus. The next asexual reproduction, and these are called the pycnospores, PYCNIO spores, pycnospores. These are conidia-like structure, just like conidia structures, but they are developed in a flask-shaped structure that is called pycnidium. A flask-shaped structure is developed. Inside you can find number of chains like conidia-like spores are there. They will be removed and liberate and form into new mycelium. This you can see here. So see, look here, it is a bowl, the structure, or a flask-like structure. This is called pycnidium. Inside the pycnidium you can have the pycniospores, or those pycniospores are developed and they will be removed and germinate into new cells. Regarding the sexual reproduction is the function of fungal partner only. Even the sexual reproduction also the function of the fungal component, whether it is Ascomycetes or Basidiomycetes. Here the male sex organ is called the spermogonium, which is a flask-shaped structure, shaped pycnidium-like structure. There you can find inside this a number of non-motile spermatia are developed. You can see the photograph later, and it is just like a couplet pycnidium-like structure, and inside you can find a number of spermatia are developed. They are released out through an opening is there. Through the opening they are released out. And the female sex organ is known as the carpogonium, which is having a coiled lower portion. It is just like a coiled lower portion is there, and having a long terminal structure. This is called the trichogyne, and inside you can find a female gamete, and these spermatia which are released out, they may be attached with that trichogyne and enter into it and fuses with the female gamete and form a dikaryotic mycelium, or a diploid zygote is formed. This diploid zygote gives a number of filaments, number of hyphae. There they unite together and produces a diploid ascus-like structure, and this is a diploid ascus-like structure undergoes meiosis and form ascospores. Each ascus gives 8 ascospores, and these 8 ascospores are inside the ascus. So this is called endogenous origin. Endogenous origin of the ascospores. So each ascus gives 8 ascospores, and all the 8 ascospores are inside the ascus. The origin is endogenous origin. After that they are released out and develop into that particular fungus. So here, as in that since it is a fungus partner, the reproduction and fruiting body is also related to this fungus only. These ascogenous hyphae, ascus mother cells, and ascospores, and again this number of cells, sterile cells, form a cup-like structure, open cup-like structure. This is called apothecium, apothecium-like structure. And after formation of these spores, they are released out and develop into the particular thallus. This type of apothecium is found in the lichen like Parmelia and Asnia. Next you see the reproduction. In some lichens the fruit body is perithecium. Perithecium is a flask-shaped structure. They are having a long flask-shaped structure and having an opening. This is called the ostiole. Through the opening, these spores are released out. The ascospores are released out. So depend upon the shape only it is given apothecium and perithecium. This type of perithecium is found in acrocardia. Acrocardia, this lichen, you can find this perithecium, reproductive structure or fruiting body. These ascospores are liberated from the ascus and germinate to give new fungal hyphae. These hyphae, in contact with the suitable algal partner, they constitute a new lichen thallus. This line is very, very important because they produce, the sexual reproduction is only done by the fungal partner. So the fungal partner gives the fungal spores. They germinate and give the fungal hyphae only. Those fungal hyphae find a suitable algae and form this lichen association. Now let us see the economic importance of lichens. So everywhere, right from this bacteria, we studied the harmful aspects and beneficial aspects, and here also we can find both beneficial as well as harmful aspects, but some interesting things also you can find the lichens. The first one, food. Several lichens are used as food. Example, Cladonia rangiferina. This is called a reindeer moss. Is used as food and fodder in tundra regions. So most of these tundra reindeers etc. are fed by this Cladonia rangifera, so that it is called a reindeer moss. And Citraria islandica, this is called island moss, is also used as food in island areas. For example, Avernia, Parmelia, etc. are used as food or fodder. Fodder means food for cattle. Fodder means food for cattle. So these species may be taken as human beings or animals also. Next, you see the medicine. So a number of medicines also extracted from this lichenic acid, because these lichens having a number of acids, a variety of acids, those are unique to lichen only. There where you cannot find, you can find only in lichens. In such case, for example, one is the arsenic acid is produced by many lichens like Arsenia, Cladonia, Lobaria, etc. It is a broad spectrum antibiotic. Broad spectrum means they can kill a variety of infections. Whereas a narrow spectrum means it kills only some particular diseases only, whereas a broad spectrum antibiotic can cure a number of or a variety of bacterial infections. So is against to the gram-positive bacteria. The next you have this erythrin, is a chemical substance obtained from lichens from Roxella montagni, used in the treating of severe heart diseases. Severe heart diseases, this medicine is used, obtained from Roxella. And species of Avernia, Lobaria are used to cure cough, fever and hydrophobia, that is a phobia towards water, a fear towards water, that is a disease that is called hydrophobia, for which these medicines are used. The next is the dyes. Dyes are nothing but the coloring materials. I think this term so far we have not used it, but now we are using. The dyes are the coloring materials obtained from particular lichens here. They are also obtaining from the plants, the higher plants that you can learn in the study of taxonomy etc. You can learn more plants and dye yielding plants. Now here, these are the dyes obtained from the lichens. The dyes are generally used to give color to the cloth. It may be a silk or cotton, and also used in paints, and also in the coloring the medicines also. Sometimes you can find some medicines are red color, yellow color, that is due to the coloring of that dyes. In this case, a purple dye, a red dye is obtained from Acrolachia species. That dye, a purple dye, a red dye is obtained from Acrolachia species, and an yellow dye is from Parmelia, and Ochil, a blue dye, is obtained from Citraria, Leconera, etc., used as a biological stain. Biological stain means generally you are taking sections and giving some stain and wash slowly and sensibly through microscope. So in the same way, to color the biological items or biological spotters etc., this dye is used. Archil is used to stain the biological organs. And the next, this archil is obtained from Citraria and Lechnor etc. is this biological stain. I already told you. The another one is very, very important to get a litmus paper. So generally the litmus paper is used in the chemistry lab to change acid to base and base to acid. That is obtained from this lichen Roxella tinctoria. Tinctorous means blue color. Tinctorous means blue color. So we can get a blue color from this litmus, that is Roxella tinctoria, used as a acid base indicator. Regarding perfumes and cosmetics, since the lichens are having an excellent smell and flavor, and also you can get a number of essential oils also, so that they are used to prepare the perfumes and use in cosmetics, soaps, etc. These are the Sudernia and Evernia are the examples from which we can obtain these essential oils and also perfumes and cosmetics. Regarding alcohols, so some lichens like Asnia, Ramelina, etc. are used in the preparation of alcohols. This is called not Ramelina, this is Ramalina, M-A-L-I-N-A, Ramalina, etc. are used in the preparation of alcohols. Regarding the economic importance, the ecological importance, there is another important thing regarding the biological weathering. Biological weathering means biologically the soil is preparing, changes in the environment. Lichens are regarded as pioneers of ecological succession on rocks and barren lands. This ecological succession is of two types. One is called xerosear, another is called the hydrosear. This xerosear is development of land, development of soil from the rocks is called xerosear, and development of vegetation from water is called hydrosear, but now it is not concerned. It is the xerosear, so the development of the land and development of vegetation from the rocks from these lichens only. So they produce a number of organic acid, I told you already. They produce a number of alkalic acids like carbonic acid and oxalic acid etc. They gradually dissolve and disintegrate the rocks over which they grow. Where you can find this lichen, they produce these acids and dissolve and disintegrate the soil particles and made some soil. Whenever they made some soil, the foliose and fructose lichens will establish there. So they produce a number of organic acids like carbonic acid and organic. They gradually dissolve soil particles and they disintegrate the soil particles, they grow. The soil is formed, that is called pedogenesis. Pedo means soil, land, that is called pedology. Pedology, study of land, is called pedology. So formation of the soil, pedogenesis. Genesis is formation, pedo means land, land genesis, in which they pave way to grow other plants subsequently. So whenever the first plants are the lichens and they dissolve the soil particles and prepare some soil, there the lichens establish again. After that, still they produce acids, still more soil is prepared, then mosses, bryophytes will develop. After bryophytes, pteridophytes will develop, and then gymnosperms, and then angiosperms, and whole vegetation is developed on the rocks. That is called the xerosear. So subsequently what happened, the sequence of different stages in xerosear, the first pioneer is the crustose lichen, followed by the foliose lichen, and they produce sand or soil, then mosses will develop, and then small herbs and the shrubs, and gradually pteridophytes, gymnosperms, angiosperms, and trees are developed. That is the climax of the xerosear. So pioneer the lichens and climax the very big trees. This is a sequence in the xerosear. the development of vegetation on barren rocks, etc. That is called erosive. So this a very good photograph. You can see these diagrams. The starting lichens start on this and gradually gives the way to the mosses, and then herbs will develop, and the shrubs will develop, and large shrubs will develop, and again you can have the large trees are developed. So this is a process of xerosil. Another very important thing in these lichens is they are the indicators of air pollution. Indicators of air pollution, because they are highly sensitive. There you can find a pollution is there, they never develop there. So you can find out that in the where you can find lichens, that area is free of pollution. If any little pollution of carbon dioxide or sulfur dioxide is there, there is no growth of these things, or sometimes they show very stunted growth, small growth or little growth, etc. And sometimes what happens, they accumulate the trace metals induces, where in atmosphere where you can find the metals are there, and they used to follow what is called they have to what's called reduce this one and accumulate those metals in their hyphae or in their what's called the plant body. So they are able to accumulate a high amount of trace metals reduce, then grow in heavy metal contaminated environment. So after testing, isolating or observing the particular lichen, they can isolate the metals and find out in a particular area what are the metals or residues are found in the atmosphere, depend upon these lichens. So they are the best indicators of air pollution. And so far we discussed only beneficial, some of the beneficial aspects. Now you see some what's called the harmful aspects of these lichens. The first one is the skin diseases. Skin diseases. Some lichens like Asnia, Avernia, they cause skin diseases and respiratory allergy, because they produce this Isidia and Oidea, etc. They give some allergy, and also they enter into the nose due to respiration and they cause allergy. So this skin allergy and respiratory allergies, and this is the skin diseases. Some lichens like Asnia and Avernia cause skin diseases and respiratory allergy. I told you already, in the skin you see it is a leg of a person where you can find the patches are found on the skin. This is due to the lichen spores or lichens. Next one, poisonous lichens, that is Citraria juniperina and Litharia pulpina, etc. are highly poisonous, or by mistake any animal used to eat those plants along with other grasses, etc. they used to die, so they are highly poisonous. Another aspect, that is the last aspect in this regarding the harmful thing, the forest fire. See, during hot summer days what happen, the lichens of foliose, fructose, etc. they become dry and inflammable, so that which catch fire from bottom to top of the trees of the forest. The whole forest will be fired because of these lichens, especially these foliose and fructose type. They are very dry and they catch fire easily and spoil the whole forest. This you can see here, because of these lichens, firing, it may be started from particular place and fire, throughout the forest will be fired due to these lichens. Okay, so far we discussed lichen, its meaning, association, and thallus structure, classification, and vegetative propagules, reproduction, vegetative reproduction, asexual reproduction, sexual reproduction, and regarding the economic importance, the beneficial aspects and also the harmful aspects. Now we will go to the next branch, that is Bryophyta. So far we discussed the what's called these bacteria, viruses, algae, fungi, lichens. So this is the next stage. Next stage, these plants from water to the land. So these are the first terrestrial plants moving from water to the land. So this Bryophyta, the term Bryophyta means bryon means mass and phyton means plant. So mass like plants. Masses, generally the Bryophyta are called masses general, and the pteridophyta are called the ferns. It's a common name, but there are much differences there inside. So these are mass like plants. The word is coined by Braun, B-R-A-U-N, in 1864. They are simple group of terrestrial non-vascular plants. As I told you already, terrestrial means land plants, but non-vascular, no vascular tissue, no xylem and no phloem. And the study of Bryophyta is known as bryology. Bryology is the name of the subject. This group is commonly known as amphibians of the plant kingdom. Amphibians, another very important thing that you remember, Bryophyta and amphibian plant kingdom comes together. There you can find these plants grow on land, but they need a moist place. They need a moist place because of this reproduction, for the fertilization, etc., water is essential. So without water they cannot complete their life cycle. So these are called amphibious plants. That is, they need land as well as water to complete their life cycle. That is called amphibians of the plant kingdom. And then Professor Sivaram Kashyap is the explorer of Himalayan bryophytes, hence called father of Indian bryology. Father of Indian bryology. So Professor Sivaram Kashyap is the explorer of the Himalayan bryophytes, hence he called the father of Indian bryology. Next general characters you can see, this one, they are inhabit of damp, shaded and humid localities. A bryophyte can live only in the humid and damp area only you can find the growth. The next one is, in the life cycle of bryophytes there are two generations, gametophytic haploid generation and sporophytic diploid generation. The gametophytic plant is conspicuous. You can see bryophyte as a what's called gametophyte only. It is a long-lived dominant plant, the gametophytic partner. And the plant body may be thalloid. So here you can find ranges from thalloid to the tree-like or plant-like. The thalloid nature you can find in liverworts, for example Erixia, are differentiated into stem-like, leaf-like and root-like structures, because they are not true leaves, they are not true roots, true stem. So they are like structures. That's why a stem-like structures. Example, the mosses you can find in Funaria. I have given only one or two examples, will be later. So mosses example, Funaria. Regarding the next characteristics, here we can see the Erixia, where you can find a thallus nature, you can find Erixia. And here you have a Funaria, where you can find here a root-like structure inside and a leaf-like structures are there, and you can find this is the sporangium that I will tell you later. This is the sporophytic generation. This is the gametophytic generation. So the sporophytic generation depend upon the gametophytic generation. Next, plant thallus. This thallus organization is very simple and no distinct root system, but they are attached to the substratum by means of thin filamentous outgrowths of the thallus called the rhizoids. They are just like root-like structures. They may be unicellular or multicellular, or in addition to these rhizoids, some scales are also found for protection purpose. The vascular tissue is absent. Here you can find no xylem, no phloem. The vascular tissue is absent. And the photosynthetic pigments, because they are green in color, photosynthetic pigments are chlorophyll A and chlorophyll B, beta carotene and xanthophylls. The reserve food materials are in the form of starch and small amounts of fats. The cell wall is made up of cellulose and hemicellulose. So this is the thallus structure and cell structure. Regarding this reproduction, they multiply vegetatively by means of tubers, gemmae or protonema. Here you can find these structures. Here you can find these are the tuber-like structures. They may be removed and develop into a new plant. And here the gemmae, here you see the flower-like structures, they are gemmae. They may be removed and develop into a new plant. And these are the protonema that you can find in mosses, where the haploid spore germinate to give a filament-like structure, and these filaments also having some buds. These buds are removed and develop into the new plant. Asexual reproduction is completely absent in this. The sexual reproduction is oogamous, the fusion of the egg with the male gamete. Fusion of the egg with male gamete. And male sex organ is known as the antheridium, and female sex organ is known as archegonium. Sex organs are haploid, multinuclear, and they have a protective jacket of a layer of sterile cells. This is the difference between algae and Bryophyta. In algae, the reproductive cells, antheridium or archegonium, do not have any outer sterile jacket. But whereas in Bryophyta, they have a protective jacket layer of sterile cells. This is the difference of algae and Bryophyta. This antheridium, that is the male reproductive structure, is club-shaped or spherical-shaped and produced by flagellate anthrozoids. So this antheridium produces biflagellate anthrozoids or male gametes. Anthrozoids are nothing but the male gametes. Archegonium, more or less, it is a flask-shaped structure. It is a flask-shaped structure and having a broad venter and a narrow neck is there at the upper side. And here you can see this is the antheridium. It is found here protected by these hyphae, these filamentous-like structures, and inside you can find antheridium, and inside you can find anthrozoids. The anthrozoids are removed out and move in the water. This is the archegonium. Narrow neck, broad venter is there, and here you can find neck cells, and in between neck canal cells, and here in this venter you have two more cells. One small cell is called the venter canal cell, and below which you have a very big egg cell. So then these male gametes moving in the water currents and move and go to this archegonium. So a number of male gametes enter into the archegonium neck, and finally one male gamete fuses with the egg and form the zygote. So depend upon the genera, the neck contains 1 to many neck canal cells. The venter encloses two cells, a small venter canal cell and a large egg cell. Fertilization takes place in the presence of water. The zygote is diploid, which develops into embryo. So the word embryo only just you learned so far. So these plants are embryophytes. The plants having embryo are called embryophytes. Embryo is formed after gametic fusion. So the embryophytes are Bryophyta, Pteridophyta, Gymnosperms and Angiosperms. These are called embryophytes. So embryo develops from here only, and the zygote represents the first cell of the sporophytic generation. So far we discussed the gametophytic generation. After the formation of the zygote, the sporophytic generation starts. The sporophyte is a complex body known as the sporogonium. The sporogonium consists of foot, seta and capsule. Three structures we can find in this, but some species having some structures, some having some other structures. So the lower portion is called the foot, from which it absorbs food material from the gametophytic plant. And this is a seta, the stalk-like structure. Above you can find a capsule. This is the proper sporogonium. So the capsule of sporogonium is the spore producing body. So as it is a fusion of male and female gametes, so sporophyte is diploid. So inside you can have all diploid cells and they form into spore mother cells. Spore mother cells are diploid. So these diploid spore mother cells undergo reduction division and form haploid spores. This sporophyte is partially or totally dependent upon the gametophyte for its nutrition. The diploid spore mother cell undergo meiosis and produce haploid spores. Another important thing here you have to remember, in Bryophyta, all the spores are of same size and structure, so this is homosporous condition. Whereas in Pteridophyta, you can find both homosporous and heterosporous. But in Bryophyta is concerned, here you can find homosporous condition. The haploid spore represents the first cell of the gametophytic generation. Then bryophytes show alternation of generations. That is, the haploid gametophyte and diploid sporophyte, they are morphologically dissimilar, different shape, so different size, different structure. So the life cycle is called heteromorphic alternation of generation. Alternation of generation means the haploid gametophyte gives diploid sporophyte and diploid sporophyte in turn gives haploid gametophyte, that is alternation of generations. In this alternation of generation, both gametophyte and sporophyte are of same size and structure, that is called isomorphic alternation of generation. But whereas here there is a difference between this gametophyte and sporophyte, so it is known as heteromorphic alternation of generations. Now let us see the classification. As I told you now, one example Rixia I have given here, and it produces the sex organs. Male sex organ antheridium, female archegonium. Archegonium gives the egg, haploid, anthrozoids. This antheridium gives anthrozoids and haploid, and fertilization takes place. A diploid zygote is formed. This diploid zygote develops into sporophyte. Sporogonium is developed. There you can find spore mother cells. Those spore mother cells undergo reduction division, meiosis, and form haploid spores. These haploid spores germinate to give the haploid gametophyte. So haploid gametophyte gives the gametes, fusion give diploid sporophyte. Diploid sporophyte gives spore mother cells, undergo reduction division and form spores, again give this gametophyte. This is called alternation of generations. So tomorrow we'll discuss the classification and the different classes. Thank you.